Hi everyone. This time my video's back, so hopefully this it'll be better if you can see my face. All right, for this video, we're just going to do page 3, which is the different stages of meiosis. The first thing I want to talk about is haploid or diploid. So if we take a look at this model of meiosis, we are starting with a diploid cell in interphase. We have a pair of homologous long chromosomes and a pair of homologous short chromosomes. So our cell, when we are starting meiosis, is diploid. Okay, now if we continue on, is our cell, now we have replicated our DNA, okay, but is our cell still diploid? Yes, it is. If I show you a picture of what our cell, our karyotype looks like in prophase, it's something like this. So we've replicated our DNA, but we still have 2 of each chromosome, okay? So it is still diploid. Now, if we continue on, it'll still be diploid, long, long, short, short, long, long, short, short, long, long, short, short, long, long, short, short. It's diploid up until cytokinesis, when we actually split those cells. So after we split those cells, now we only have one long, one short. One long, one short, because we have split our homologous chromosomes. So up until cytokinesis or prophase 2, our cell is diploid, and then it becomes haploid. All right. Now if we go back to our model, right, and we go over here to meiosis 2, take a look at it. I'm sorry, my computer's lagging a little bit. Is my cell in prometaphase and in metaphase haploid? Yes, it is. One short, one long. Okay. However, here in anaphase 2, is it haploid? No, because my sister chromatids have split. And once again, I have 2 short chromosomes and 2 long chromosomes. Even in telophase, I have 2 short and 2 long. Okay. However, once cytokinesis happens and these cells fully split, my gametes are once again haploid. Okay, sorry, I think it's because I'm uploading the other YouTube video I made. Okay, so we said in meiosis, in metaphase 2, my sister chromatids are still connected, so my cell is still haploid. However, once those sister chromatids are splitting in anaphase 2, then we have a diploid cell temporarily in anaphase 2 and telophase 2. But then, once it fully splits in cytokinesis, then we have our haploid gametes, okay? If we describe what's going on in each of these stages, I'm going to say the most important thing about interphase is that in interphase, we have replication of DNA, okay? So our chromosomes are replicating. I'm going to say metaphase 1, what is important about metaphase 1? This is when we have crossing over between homologous chromosomes. Anaphase 1 and telophase 1, this is when our homologous chromosomes are splitting, okay? So this is different than anaphase 2 because in anaphase 2, what is splitting? Our sister chromatids are splitting. If that is confusing to you, please go back to the first page and look at your goals for meiosis 1 and meiosis 2. Cytokinesis, this is our first split into 2 daughter cells. We have prophase 2, not too much exciting there. Metaphase 2, oh, I should say, what is occurring in anaphase when they're splitting? Independent assortment, right? I'm sorry. Should have put independent assortment here. And also in anaphase 2, we should have our independent assortment, right? Okay. Prophase 2, most important thing here is that this is haploid, right? Metaphase 2, our chromosomes are aligning at the equator of the cell, or you can call it the middle of the cell. Okay. And most important thing about our gametes is that it results in 4 genetically different haploid gametes. Okay. That is the result of our meiosis. Draw a Venn diagram comparing and contrasting meiosis and mitosis. I'm not going to do this because it should already be in your notebook, okay? We very clearly did this in the notebook. If you don't have it, then you can get it from someone else if you were absent or something like that. But we went through it. I mean, I guess I can go through some features quickly. If you already have it in your notebook, you can kind of skip it, right? Okay, I'll just do it quickly. Mitosis, it results in what? Two genetically identical daughter cells. This is different than meiosis because meiosis results in 4 genetically different daughter cells / gametes. Okay. Mitosis, our daughter cells are identical to the parent cell, whereas in meiosis, the gametes are genetically different than the parent cell that we started with, right? Mitosis, it occurs in and produces somatic cells. For what purpose? For growth and repair of an organism. And meiosis, it occurs in germ cells. This is what we're starting with. And produces gametes to pass on genetic information. Mitosis, our cell only goes through one splitting, okay? Whereas in meiosis, our cell splits twice. Because we are splitting our cell twice, our gametes, our resulting gametes, are haploid. Whereas in mitosis, the resulting gametes are diploid. I'm sorry, not resulting gametes, resulting cells. Okay, daughter cells. Oh my gosh. Sorry, it's late. Resulting daughter cells are diploid. Okay, these are some of the differences. Also, meiosis, we have crossing over occurs. All right. And I think that's good. Bye, guys.